बायोटेक्नोलॉजी का ये एनसीईआरटी है जिसमें हम देखेंगे बायोटेक्नोलॉजी प्रिंसिपल एंड प्रोसेसेस जो कि हमारा फर्स्ट चैप्टर रहेगा, तो अभी है जो इसका एनसीईआरटी देखेंगे। बाद में बायोटेक्नोलॉजी एंड इट्स एप्लीकेशन का वाला पार्ट देखेंगे। तो इंट्रोडक्शन के रूप में एनसीईआरटी कह रहा है कि एवर सिंस द डेज ऑफ द रेनी डिस्कार्ड्स द फ्रेंच फिलोसफर मैथमेटिशियन एंड द बायोलॉजिस्ट of the 20th century. All human knowledge, especially the natural science, were directed to develop technology which add to the creature comforts of the human life as also value to the human life. The whole approach to the understanding natural phenomena became anthropocentric. Physics and chemistry gave rise to the engineering, technology, industry, which all worked for the human comfort and the welfare. The major utility for the biological world is as a source of the food. Biotechnology, the 20th century of food of the modern biology changed our daily life as its product, brought qualitative improvement in the health and the food production. The basic principles underlying the biotechnological processes and some applications are highlighted and discussed in this unit. तो हार्बर्ट बोयर के बारे में कुछ दिए हुए यहाँ पर लिखा हुआ है कि हार्बर्ट बोयर was born in the 1936 and brought up in a corner of the western Pennsylvania where railroads and the mines were the destiny of most young men. He completed graduation work at the University of the Pittsburgh in 1953, followed by the 3 years of the postgraduate study at the Yale. In 1956, Boer took over assistant professorship at the University of the California at San Francisco. By 1969, he performed a study on a couple of the restriction enzyme RE of the E. coli bacterium with especially useful properties. Boyer observed that these enzymes gave the capability of cutting DNA strands in a particular fashion, which left heart batch become known as the sticky ends on the strands. These clipped ends made pasting together pieces of the DNA a precise exercise. This discovery in turn led to a rich and rewarding conversation in the Hawaii with a Stanford scientist, namely the Stan Cohen. Cohen had been studying small ringlets of the DNA called the plasmid, and which float about freely in the cytoplasm of the certain bacterial cells and replicate independently from the coding strand of the DNA. Cohen had developed a method of removing these plasmids from the cell and then reinserting them in the other cells. Combining this process with that of the DNA splicing enabled Boyer and the Cohen to recombine fragments of the DNA in the desired configuration and insert the DNA in the bacterial cells, which could then act as a manufacturing plant for specific proteins. This breakthrough by the basis upon which the disciplines of the biotechnology was founded. तो ये है बायोटेक्नोलॉजी प्रिंसिपल एंड द प्रोसेसेस, जिसमें हम देखेंगे प्रिंसिपल्स ऑफ द बायोटेक्नोलॉजी का टॉपिक, टूल्स ऑफ द रिकॉम्बिनेंट डीएनए टेक्नोलॉजी और प्रोसेस ऑफ द रिकॉम्बिनेंट डीएनए टेक्नोलॉजी का टॉपिक। तो एनसीईआरटी स्टार्ट ऐसे करता है कि बायोटेक्नोलॉजी डील्स विद द टेक्निक्स ऑफ यूजिंग लिव ऑर्गेनिज्म और द enzymes from the organism to produce the products and the process is useful to the humans. In this sense, making curd, bread or the wine, which are all micro-mediated processes, could also be thought as a form of the biotechnology. However, it is used in a restricted sense today. We refer to such of these processes which use genetically modified organisms to achieve the same on a larger scale. Further, many of other processing techniques are also included under the biotechnology. For example, in vitro fertilization leading to the test-to-baby synthesis, synthesizing a gene and using it, developing a DNA vaccine or correcting a defective gene, all are the part of the biotechnology. The European Federation of the Biotechnology (EFB) has given a definition of the biotechnology that encompasses both traditional view and the modern molecular biotechnology. The definition given by the EFB is follow: the integration of the natural science and the organism, cell parts thereof, and the molecular analogs for the products and the services. About the principles of the biotechnology, तो कहता है कि among many the two core techniques that enabled birth of the modern biotechnology are the genetic engineering technique to alter the chemistry of the genetic material DNA and the RNA, to introduce these into the host organism and thus change the phenotype of the host organism. फिर आता है bioprocess engineering, तो कहता है maintenance of the sterile, microbial, contamination-free ambience in the chemical engineering process to enable the growth of only the desired microbe or eukaryotic cell in the large quantity for the manufacture of the biotechnological products like the antibiotic, vaccines, enzyme, etc. फिर कहता है कि Let us now understand the conceptual development of the principle of the genetic engineering. Kata, you probably appreciate the advantage of the sexual reproduction over the asexual reproduction. The former provides the opportunity for the variation and the formulation of the unique combination of the genetic setup, some of which may be beneficial to the organism as well as the population. Asexual reproduction preserves the genetic information while the sexual reproduction permits the variation. Traditional hybridization procedure used in the plant and the annual breeding very often lead to the inclusion and multiplication of the undesirable genes along with the desired genes. The technique of the genetic engineering, which include the creation of the recombinant DNA, use of the gene cloning and the gene transfer, overcome this limitation and allow us to isolate and introduce only one or a set of the desired genes without introducing undesirable genes into the target organism. फिर कहता है कि डू नो द लाइकली फेट ऑफ द पीस ऑफ द डीएनए विच इज समहाउ ट्रांसफर्ड इनटू एन एलियन ऑर्गेनिज्म मोस्ट लाइकली दिस पीस ऑफ द डीएनए would not be able to multiply itself in the progeny cells of the organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of the DNA has become part of the chromosome, which has the ability to replicate. In chromosome, there is a specific DNA sequence called the origin of the replication, which is responsible for initiating the replication. Therefore, for the replication of any alien piece of the DNA in an organism, it needs to be a part of the chromosome which has a specific sequence known as the origin of the replication. Thus, an alien DNA is linked with the origin of the replication so that this alien piece of the DNA can replicate and multiply itself in the host organism. This can also be called as the cloning or making multiple identical copies of any template DNA. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid autonomously replicating circular extrachromosomal DNA of the Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistant gene by cutting out a piece of the DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of the DNA at a specific location became possible with the discovery of the so-called uh, molecular seizure restriction enzyme. The cut piece of the DNA by then linked with the plasmid DNA. This plasmid DNA act as a vector to transfer the piece of the DNA attached to it. You probably know that the mosquito act as an insect vector to transfer the malarial parasite into the human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of the DNA into the host organism. The linking of the anti the Linking of the antibiotic resistant gene with the plasmid vector become possible with the enzyme DNA ligase, which acts on the cut DNA molecule and join their ends. This makes a new combination of the circular autonomously replicating DNA created in vitro and is known as the recombinant DNA. When this DNA transferred into the E. coli, a bacterium closely related to the Salmonella, it could replicate using the new host DNA polymerase enzyme and make the multiple copies. This ability to multiply a copy of antibiotic resistant gene in E. coli was called the cloning of the antibiotic resistant gene in the E. coli. You can hence infer that there are three basic steps in the genetic modifying an organism. Pehla, identification of the DNA with the desired genes. दूसरा, introduction of the identified DNA into the host. और तीसरा, maintenance of the introduced DNA in the host and transfer of the DNA to its progeny. फिर आता है tools of the recombinant DNA technology. तो कहता है कि now we know from the foregoing discussion that the genetic engineering or the recombinant DNA technology can be accomplished only if we have key tools, that means the restriction enzyme, polymerase enzyme, ligase, vectors and the host organism. Let us try to understand some of these in the detail. तो पहला आता है रिस्ट्रिक्शन एंजाइम. कहता है कि इन द ईयर 1953 द टू एंजाइम रिस्पॉन्सिबल फॉर रिस्ट्रिक्टिंग द ग्रोथ ऑफ द बैक्टीरियोफाज इन द Escherichia coli were isolated. One of these added methyl groups to the DNA while the other cut DNA. The latter was called the restriction endonuclease. The first restriction endonuclease HIN2, whose functioning depend on a specific DNA nucleotide sequence, by isolated and characterized by 5 years later. It was found that HIN2 always cut DNA molecule at a particular point by recognizing a specific sequence of the 6 base pair. This specific sequence is known as the recognition site for the HIN2. Besides HIN2 to be no more than now 4 restriction enzymes that have been isolated from over 230 strains of the bacteria, each of which recognize different recognition sequence. The convention for naming these enzymes is the first letter of the name comes from the genus and the second two letters come from the species of the prokaryotic cell from which they were isolated. E core 1 comes from the Escherichia coli, R by 1 3. In the E core 1, the letter R is derived from the name of the strain. Roman numerals following the name indicate the order in which the enzymes were isolated from that strain of the bacteria. फिर कहता है restriction enzyme belong to a larger class of the enzyme called the nuclease. These are of two kinds, exonuclease and the endonuclease. Exonuclease remove the nucleotide from the ends of the DNA, whereas endonuclease make cuts at the specific position within the DNA. फिर कहता है each restriction endonuclease function by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at a specific points in their original sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in this DNA. तो दिया हुआ है steps in the formation of the recombinant DNA by the action of the restriction endonuclease enzyme ECOR1. इसमें दिया हुआ है enzyme action of the restriction enzyme के बारे में दिया है तो enzyme cuts both DNA strands at the same site पर, ये vector DNA हो गया. ECOR1 cut the DNA between the bases G and A only when the sequence G80TC is present in the DNA. तो foreign DNA हो गया. ECOR1 कट किया इस तरह से कि स्टिकी इंड बन गए हैं और जिग जैग कट किया है तो DNA फ्रैगमेंट स्टिकी इंड पर ज्वाइन हो जाएगा और रिकॉम्बिनेंट DNA जो बन जाएगा लाइजेज के द्वारा है जो ज्वाइन किए हैं. फिर कहता है कि डू नो व्हाट पैलिंड्रोमिक आर. दीज आर द ग्रुप ऑफ द लेटर्स दैट फॉर्म द सेम वर्ड व्हेन रीड बोथ फॉरवर्ड एंड द बैकवर्ड. जैसे मलयालम एज अगेंस्ट ए वर्ड पैलिंड्रोमिक बेयर द सेम palindrome where the same word is read in the both direction. The palindrome in DNA is a sequence of base pair that reads same on the two strands when orientation of the reading is kept in the same. For example, the flank sequence reads the same on the two strands in the 5-3' direction. This is also true if we read in the 3-5' direction. जैसे 5-3' में GATTC और 5-3' में GATTC उल्टा हो जाएगा. ये हो गया ECOR 1 के लिए. फिर आता है कि restriction enzyme cut the strands of the DNA a little away from the center of the palindrome sites but between the same two bases on the opposite strands. This leaves single strand portion at the ends. There are overhanging stretches called the sticking ends on each strand. These are named so because they form the hydrogen bonds with their complementary cut counterparts. This stickiness of the ends facilitates the action of the enzyme DNA ligase. Restriction endonucleases are used in the genetic engineering to form the recombinant molecule of the DNA, which are composed of the DNA from the different source of the genome. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of the sticking ends, and these can be joined together end to end using the DNA ligase. अब यहाँ पर दिया हुआ है डायग्रामेटिक रिप्रेजेंटेशन ऑफ द रिकॉम्बिनेंट डीएनए टेक्नोलॉजी. इसमें ये फॉरेन डीएनए हो गया, फिर वेक्टर डीएनए प्लाज्मिड हो गया और सेम रिस्ट्रिक्शन एंजाइम कटिंग बोथ फॉरेन डीएनए एंड द वेक्टर डीएनए एट अ स्पेसिफिक साइट पर. लैगेन ज्वाइन द फॉरेन डीएनए टू द प्लाज्मिड और रिकॉम्बिनेंट डीएनए मॉलिक्यूल हो गया जो ट्रांसफॉर्मेशन हो रहा है. ये कोलाई क्लोनिंग होस्ट और द सेल है जो डिवाइड कर रहे हैं. फिर कहता है कि you may have realized that normally unless one cut the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Separation and isolation of DNA fragments. तो कहता है the cutting of the DNA by the restriction endonuclease results in the fragments of the DNA. These fragments can be separated by a technique known as the gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under an electrical field through a medium and matrix. Nowadays, the most commonly used matrix is agarose, which is natural polymer extracted from the seaweeds. The DNA fragments separate resolve according to their size through the sieving effect provided by the agarose gel. Hence, the smaller fragment size, the farther it moves. Look at the figure 9.3 and guess at which end of the gel the sample was loaded. फिर कहता है कि the separated DNA fragments can be visualized only after staining the DNA with a compound known as the ethidium bromide followed by the exposure to the UV radiation. You cannot see the pure DNA fragments in the visual light, and without staining, you can see the bright orange colored bands of the DNA in ethidium bromide of standard gel exposed to the UV light. The separated bands of the DNA are cut out from the agarose gel and extracted from the gel piece. This step is known as the elution. The DNA fragments purified in this way are used in the constructing recombinant DNA by joining them with the cloning vectors. यहाँ पर दिए हुए है a typical agarose gel electrophoresis showing migration of the undigested lane 1 and digested set of the DNA fragments lane 2 to 4. तो इधर बेल्स हो गया, लार्जेस्ट इधर, स्मॉलेस्ट है जो सबसे आगे रहेगा. फिर एनसीईआरटी क्लोनिंग वेक्टर्स के बारे में कहता है कि यू नो दैट प्लाज्मिड एंड द बैक्टीरियोफेज हैव द एबिलिटी टू रिप्लिकेट विद इन द बैक्टीरियल सेल्स इंडिपेंडेंट ऑफ द कंट्रोल ऑफ द क्रोमोसोमल डीएनए. Bacteriophage, because of their high number per cell, have very high copy number of the genome within the bacterial cells. Some plasmid may have only 1 or 2 copies per cell, whereas others may have 15 to 4 copies per cell. Their number can go even higher. If we are able to link an alien piece of the DNA with bacteriophage or the plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. Vectors used at present are engineered in such a way that they help easy linking of the foreign DNA and selection of the recombinants from the non-recombinants. आगे एनसीईआरटी कहता है कि द फॉलोइंग आर द फीचर्स दैट आर रिक्वायर्ड टू फैसिलिटेट क्लोनिंग इनटू ए वेक्टर. पहला ऑरिजिन ऑफ रेप्लिकेशन तो कहता है दिस इज अ सीक्वेंस फ्रॉम वेयर रेप्लिकेशन ऑफ स्टार्ट्स एंड एनी पीस ऑफ डीएनए विल लिंकेड टू दिस सीक्वेंस कैन बी मेड टू रेप्लिकेट विद इन द होस्ट सेल. This sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. फिर आता है फ्लेक्टिबल मार्कर तो कहता है इन एडिशन टू ORI द वेक्टर रिक्वायर्स अ फ्लेक्टिबल मार्कर विच हेल्प्स इन आइडेंटिफाइंग एंड एलिमिनेटिंग नॉन ट्रांसफॉर्मेंट एंड selectively permitting the growth of the transformant. Transformation is procedure through which a piece of the DNA is introduced in a host bacterium. You will study the process in a subsequent section. Normally in the genes encoding resistant to the antibiotics such as ampicillin, chloramphenicol, tetracycline or the chiamycin etc. are considered useful selectable marker for the E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. इसके आगे आता है क्लोनिंग साइट्स तो कहता है कि इन ऑर्डर टू लिंक द alien DNA, the vectors need to have very few, preferably single, recognition site for the commonly used restriction enzyme. Presence of more than one recognition sites within the vectors will generate several fragments which will complicate the gene cloning. The ligation of the alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. For example, you can ligate a foreign DNA at the BAM-H1 site of the tetracycline resistant gene in the vector PBR322. The recombinant plasmid will lose tetracycline resistance due to the insertion of the foreign DNA but can still be selected out from the non-recombinant ones by the plating the transformant on tetracycline containing medium. The transformants growing on the ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in the ampicillin containing medium but not on that containing tetracycline. But non-recombinants will grow on the medium containing both the antibiotics. In this case, one antibiotic resistant gene helps in selecting the transformant, whereas the other antibiotic resistant gene gets inactivated due to the insertion of the alien DNA and helps in the selection of the recombinants. Selection of the recombinants due to the inactivation of the antibiotic is a cumbersome procedure because it requires simultaneous plating on the two plates having different antibiotics. Therefore, the alternative selectable markers have been developed which differentiate recombinants from the non-recombinants on the basis of their ability to produce the color in the presence of a chromogenic substrate. In this, a recombinant DNA is shunted within the coding sequence of the enzyme beta-galactosidase. This results into the inactivation of the gene for the synthesis of this enzyme, which is referred to as the insertional inactivation. The presence of the chromogenic substrate gives the blue-colored colonies if the plasmid in the bacteria do not have an insert. Presence of the insert results into insertional inactivation of the beta-galactosidase gene and the colonies do not produce any color. These are identified as the recombinant colonies. अब आगे कहता है कि वेक्टर्स फॉर क्लोनिंग जीन्स इन द प्लांट्स एंड एनिमल्स तो कहता है यू मे बी सरप्राइज टू नो दैट वी हैव लर्न द लेसन फॉर ट्रांसफरिंग जीन्स into the plants and animals from the bacteria and virus which have known this for the ages. How to deliver genes to transform eukaryotic cells and force them to do what the bacteria or the virus want. For example, Agrobacterium tumefaciens, a pathogen of the several dicot plants, is able to deliver a piece of the DNA known as the tDNA to transform the normal plant cell into the tumor and direct the tumor cell to produce the chemicals required by the pathogen. Similarly, retrovirus and the animals have the ability to transform the normal cell into the cancerous cell. A better understanding of the art of delivering genes by the pathogen in their eukaryotic host has generated knowledge to transform these tools of the pathogen into useful vectors for delivering gene of the interest to the humans. The tumor-inducing plasmid of the Agrobacterium tumefaciens has now been modified into a cloning vector which is no more pathogenic to the plant but is still able to use the mechanism to deliver genes of our interest into a variety of the plant. Similarly, retrovirus have also been deformed and are now used to deliver desirable genes into the animal cells. So, once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial plant or animal host where it multiplies. यहाँ पर दिया हुआ है, दिखाए हुए इसमें पेस्ट एक का दिखाया हुआ है, पीवी एक है, इकोर 1 है, फिर काल एक हो गया, हिंड 2 हो गया, बाम एक हो गया, फाल एक हो गया. और फिर रोप पर पीवी 2 है और एफाइट है. तो एम्पिसिलिन पर अगर देखें तो पेस्ट एक पीवी एक आता है, फिर इकोर एक, काल एक, हिंड 3 आता है, हिंड 3 है. फिर इधर बाम एक है, टेट्रासाइक्लिन पर फाल एक है, और रोप पर पीवी एक 2 है, पीवी 2 है. इधर जितना भी एम्पिसिलिन है, टेट्रासाइक्लिन सब पर एक एक वाला है, सब पर है जो एक एक वाला है. और इकोर और काल भी एक है, लेकिन हिंद वाला है जो 3 गो हिंद है. और अगर रोप पर देखें तो 2 वाला है, पीवी 2 है. इस तरह से है. तो ई कोलाई है जो फोइंग, ई कोलाई फोइंग द क्लोनिंग वेक्टर पीवीआर 322 फोइंग द रिस्ट्रिक्शन फाइट, हिंद 3, इकोर 1, बाम 1, फाल 1, पीवी 2, पेस्ट 1, कॉल 1, और फाइट एंड द एंटीबायोटिक रेसिस्टेंट जीन, ampicillin and the tetracycline. Rope codes for the protein involved in the replication of the plasmid. अब इसके आगे एनसीईआरटी कंपोनेंट फॉर द ट्रांसफॉर्मेशन विद द रिकॉम्बिनेंट डीएनए के बारे में कहता है कि since DNA is the hydrophilic molecule, it cannot pass through cell membranes. Why? In order to force bacteria to take up the plasmid, the bacterial cells must be made competent to take up the DNA. This is done by treating them with a specific concentration of a divalent cation, such as the calcium, which increase the efficiency with which DNA enters the bacterium through the pores in its cell wall. Recombinant DNA can then be forced into such cell by incubating the cell with a recombinant DNA on the ice, followed by placing them briefly at 42 degrees Celsius heat shock, and then by putting them back on the ice. This enables the bacteria to take up the recombinant DNA. फिर कहता है this is not the only way to introduce the alien DNA into the host cell. In a method known as the microinjection, recombinant DNA is directly injected into the nucleus of the animal cell. In another method suitable for the plant, cells are bombarded with high-velocity microparticles of the gold or the tungsten, coated with the DNA in a method known as the ballistic or the gene gun. And the last method uses disarmed pathogen vector which when allowed to infect the cell, transfer the recombinant DNA into the host. Now that we have learnt about the tools for the constructing recombinant DNA, let us discuss the process facilitating the recombinant DNA technology. The process of the recombinant DNA technology कहता है कि recombinant DNA technology involves several steps in the specific sequence such as the isolation of the DNA, fragmentation of the DNA by the restriction endonuclease, isolation of the desired DNA fragments, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cell in a medium at large scale, and extraction of the desired products. Let us examine each of these steps in some detail. Isolation of the genetic material DNA. तो कहता है recall that nucleic acid is the genetic material of all the organism without exception. In majority of the organism, this is deoxyribonucleic acid or the DNA. In order to cut the DNA with the restriction enzyme, it needs to be in the pure form, free from the other macromolecules. Since the DNA included within the membranes, we have to break the cell open to release the DNA along with the macromolecules such as the RNA, proteins, polysaccharides, and also lipids. This can be achieved by treating the bacterial cells of plants or plant animal tissue with enzymes such as the lysozyme bacteria, cellulase, plant cells, chitinase, fungus. You know that genes are located on the long molecule of the DNA intertwined with the proteins such as the histone. The RNA can be removed by treatment with the ribonuclease, whereas proteins can be removed by treatment with the protease. Other molecules can be removed by the appropriate treatment, and purified DNA ultimately precipitate out after the addition of the chilled ethanol. This can be seen as the collection of the fine threads in the suspension. तो यहाँ पर दिया हुआ है DNA that separates out by removing by the spooling. स्पूलिंग करके स्पून से उसको है जो रिमूव आउट कर दिए. अब कटिंग ऑफ द DNA at the specific location, तो कहता है कि restriction enzyme digestion are performed by incubating purified DNA molecule with the restriction enzyme at the optimal condition for that specific enzyme. Agarose gel electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is negatively charged molecule, hence it moves towards the positively electrode anode. The process is repeated with the vector of DNA also. The joining of the DNA involves several processes. After having cut the source DNA, as well as the vector DNA with a specific restriction enzyme, the cutout gene of the interest from the source DNA and the cut vector with a specific are mixed and ligase is added. This results in the preparation of the recombinant DNA. तो आगे आता है एम्पलीफिकेशन ऑफ द जीन ऑफ इंटरेस्ट यूजिंग द पीसीआर। तो कहता है कि पीसीआर स्टैंड फॉर द पॉलीमरेज चेन रिएक्शन। इन दिस रिएक्शन मल्टीपल कॉपीज ऑफ द जीन और द डीएनए ऑफ द इंटरेस्ट इज सिंथेसाइज्ड इन विट्रो यूजिंग द two sets of the primer, small chemically synthesized oligonucleotides that are complementary to the regions of the DNA and the enzyme DNA polymerase. The enzyme extends the primer using the nucleotide provided in the reaction and the genomic DNA as a template. If the process of the replication of the DNA repeated many times, the segment of the DNA can be amplified to approximately billion times. That means 1 billion copies are made. Such repeated amplification is achieved by the use of the thermostable DNA polymerase isolated from the bacterium Thermus aquaticus, which remain active during the high temperature induced denaturation of the double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for the further cloning. तो यहाँ पर दिए हुए है डीनेचुरेशन जो कि होता है 95 डिग्री सेल्सियस पर, फिर एनिलिंग 50 से 70 डिग्री सेल्सियस पर, एक्सटेंशन जो कि होता है 75 डिग्री सेल्सियस पर। डीनेचुरेशन में जो है हाइड्रोजन बॉन्ड्स को है जो ब्रेक कर रहे हैं, और एनिलिंग में है जो प्राइमर आ गया, और फिर एक्सटेंशन में तक पॉलीमरेज आ जाता है जो फिर है जो नया डीएनए सिंथेसाइज करता है। तो 30 साइकिल में ऐसे करेंगे तो लगभग 1 बिलियन टाइम्स एम्पलीफाइड हो जाएगा, तो मतलब 1 बिलियन कॉपी है जो बन जाएगा। तो पॉलीमरेज चेन रिएक्शन पीसीआर इच साइकिल है 3 स्टेप: डीनेचुरेशन, प्राइमर एनिलिंग और तीसरा है एक्सटेंशन ऑफ द प्राइमर। फिर आता है इंसर्शन ऑफ द रिकॉम्बिनेंट डीएनए इनटू द होस्ट सेल ऑर्गेनिज्म। तो एनसीईआरटी कहता है कि देयर आर सेवरल मेथड ऑफ इंट्रोड्यूसिंग द लाइगेटेड डीएनए इनटू द रिसिपिएंट सेल्स। रिसिपिएंट सेल्स आफ्टर मेकिंग देम competent to receive take up the DNA parent in sounding. So if a recombinant DNA bearing gene of resistant to an antibiotic, example ampicillin, is transferred into E. coli cells, the host cell become transferred into the ampicillin resistant cell. If we spread the transferred cell on agar plates containing ampicillin only, transformants will grow and transformant resistant cell will die. Since due to the ampicillin resistant gene, one is able to select a transformed cell in the presence of the ampicillin. This ampicillin resistant gene in this case is called the selectable marker. अब आता है obtaining the foreign gene product। तो इंसर्ट कहता है कि when you insert a piece of the alien DNA into a cloning vector and transfer it into the bacterial, plant or the animal cell, the alien DNA get multiplied. In almost all the recombinant technology, the ultimate aim is to produce a desirable protein. Hence, there is a need for the recombinant DNA to be expressed. The foreign gene get expressed under the appropriate conditions. The expression of the foreign gene in the host cell involves understanding many technical details. After having cloned a gene of interest and having optimized the condition to induce the expression of the target protein, one had to consider producing it on a large scale. Can you think of any region why there is a need of large scale production? If any protein encoding gene is expressed in a heterologous host, it is called a recombinant protein. The cells of harboring cloned genes of the interest may be grown on a small scale in the laboratory. The cultures may be used for extracting the desired protein and then purifying it by using a different separation technique. The cells can also be multiplied in a continuous culture system, wherein the medium is drained out from the one side while the fresh medium is added from the other to maintain the cells in their physiologically most active log या expansion phase. This type of culturing method produces a larger biomass leading to the higher yields of the desired protein. Small volume culture cannot yield a appreciable quantity of the products. To produce in a large quantity, the development of the bioreactors where the large volume source 1,000 liter of the culture can be processed was required. Thus, the bioreactors can be thought of as a vessel in which raw materials are biologically converted into specific products, delivered enzymes, etc., using microbial plant animal for the human cell. A bioreactor provides the optimum condition for achieving the desired product by providing optimum growth condition, temperature, pH, substrate, salt, vitamins, oxygen. The most commonly used bioreactors are of string type, which are shown in the figure 9.7. तो दिया हुआ है यहाँ पर दिया हुआ है सिंपल स्टेट टैंक बायोरिएक्टर जिसमें दिखाया हुआ है एसिड बेस पीएच पर कंट्रोल स्टीम फॉर द स्टलाइजेशन के लिए मोटर दिए हुए, फोम ब्रेकर है, फिर है फ्लैट ब्लेड इम्पेलर, कल्चर ब्रोथ और इधर स्ट्राइल लेयर आता है। फिर इसमें हो गया अस्पारगेड, ये है अस्पारगेड स्टेट टैंक बायोरिएक्टर थ्रू विच द स्टेराइल एयर बबल फॉर स्पार्गेट। तो इसमें आ जाता है इंक्रीज सरफेस एरिया फॉर द ऑक्सीजन ट्रांसफर, बबल्स ड्रामेटिकली इंक्रीज द ऑक्सीजन ट्रांसफर एरिया, और इधर दिए हुए है गैस इंटरटेनमेंट। तो Ncert कहता है कि स्टीरेट टैंक रिएक्टर यूजुअली सिलिंडर और विथ कर्व्ड वेव्स टू फैसिलिटेट द मिक्सिंग ऑफ द reactor contents, and the exterior facilitate even mixing and oxygen availability throughout the bioreactor. Alternatively, air can be bubbled. Alternatively, air can be bubbled through the bioreactor. If you look, alternatively air can be bubbled through the bioreactor. If you look at the figure closely, you will see the bioreactor has an aerator system, an oxygen delivery system, and a foam control system, a temperature control system, pH control system, and sampling ports so that a small volume of the culture can be withdrawn periodically. फिर आगे आता है नेक्स्ट में डाउन स्ट्रीम प्रोसेसिंग। तो कहता है कि आफ्टर कंप्लीशन ऑफ द बायोसिंथेटिक स्टेज द प्रोडक्ट हैड टू बी सब्जेक्टेड टू ए फ्रीज ऑफ द प्रोसेस बिफोर इट इज रेडी फॉर द मार्केटिंग एज ए फिनिश्ड product. The process includes the separation and the purification, which are collectively referred to as the downstream processing. The product had to be formulated with suitable preservative. Such formulation had to undergo through the clinical trials, as in the case of the drugs. Strict quality control testing for each product is also required. The downstream processing and the quality control testing vary from the product to the product. अब समरी में आता है कि बायोटेक्नोलॉजी डील्स विथ लार्ज स्केल प्रोडक्शन एंड द मार्केटिंग ऑफ द प्रोडक्ट्स एंड प्रोसेसेस यूजिंग द लाइव ऑर्गेनिज्म cells for the enzyme. Modern biotechnology using genetically modified organism was made possible only when man learned to alter the chemistry of the DNA and construct the recombinant DNA. This key process is called the recombinant DNA technology or the genetic engineering. This process involves the use of the restriction endonuclease DNA ligase, appropriate plasmid or the viral vector to isolate and ferry the foreign DNA into the host organism. Expression of the foreign gene, purification of the gene product, that means the functional protein, and finally making a suitable formulation for marketing. Large scale production involves the use of the bioreactors. तो ये NCERT हो जाता है बायोटेक्नोलॉजी प्रिंसिपल एंड द प्रोसेसेस का।